Good morning, friends. Today my lecture is on cell and its mechanism of generation of energy. Cell, the basic unit of life. It is the smallest unit which can reproduce, which can perform its all biological activities by its own, and also it can create its energy to perform its activity by its own, and it is the smallest unit which can reproduce also, first thing. And cell is formed from certain components, certain biomolecules. So we will discuss cell in the two different classes. First, how it generates energy, and another that how, from which molecules it was formed from. So the first question is how the cell generates energy to perform its biological activities. And the second is the components of the cell. In components of the cell, we will later discuss, there are the 5 components. We named them biomolecules. These biomolecules are lipids, proteins, nucleic acid, carbohydrates, and water. But these biomolecules are non-living. But when all biomolecules combine with each other, they form cell, which can perform all the activities by its own. How? How it happens? How the cell creates energy by its own? We have to understand that mechanism. I'm explaining you this mechanism, that how the cell creates energy to perform its activities by its own. For understanding it, I just want to tell that we can take an example of dam. In case of dam, a flowing river, we want to create energy. We want to extract energy from a flowing river water. What we do? We create a wall. Due to this, the difference of water level takes place across the wall. Here, the potential energy of water is high, and here, the potential energy of water is low. And by the principle of thermodynamics, everything wants to move to the lowest energy state. So what happens? When we roll out this water and leave it to flow and create a turbine here, the energy was extracted in form of electricity. Now, if you understand this example, I will explain you that how cell uses this mechanism, this simple mechanism, to create energy for functioning of all the activities. In case of cell, if we look at the structure of the cell, how the cell is formed from? The primary cell is formed from a membrane. Membrane, which is made up of lipid biomolecules. Its nature is non-polar. It is non-polar in nature. A non-polar membrane, the external environment of is water, that is polar in nature. The internal environment is water, that is polar in nature. So it is, this membrane is like a wall. And in this case, the difference was the high proton gradient. The difference is the proton concentration across the membrane. Here, the proton concentration is high. Here, the proton concentration is low. And how the cell uses this difference to generate energy? The cell has a specific molecule that is named as ATPase. It is an enzyme. A specific molecule that is ATPase, which helps in creation of energy. The bigger structure of ATPase, this is a biological membrane, and this is an ATPase molecule. And when the protons across the membranes moves, because it is a polar molecule, when it moves from this C ring of this ATPase, these molecules rotate, alpha, beta, gamma. These are 6 molecules. These molecules rotates, and like a fan, in the similar way that the turbine moves, and ADP phosphate binds to form ATP. It is a molecule of high energy phosphate bond. High energy phosphate bond. So, it is like a turbine, it is like a wall, and the proton gradient was created across the membrane. ATP is an energy currency, and the cell generates this ATP by creating a proton gradient across a membrane. This phenomena was same in all that cases. For an example, in case of, I will taking an example of bacteria. Look at it. In a bacterial cell, a proton gradient was created across the membrane, inner and outer membrane of the bacterial cell. This specific organelle was named as mesosome in case of bacteria, and the proton gradient was created across the membrane. And when the protons moves from outer to in this gradient, the energy was generated. In case of plants, we observed a cell organelle, that is chloroplast. And what was the structure? It is the inner membrane, that is called as thylakoid membrane. And the gradient was created across this inner membrane, and ATP was generated. In case of, and we observed another cell organelle that is also called as powerhouse of a cell, that is mitochondria. that is mitochondria, and energy was created, gradient was created across this membrane and ATP was generated. ठीक है ना? That is inner mitochondrial membrane. So it is a similar mechanism in all three types of cell organelles and the bacterial cell. We observe the gradient was created across the membrane and ATP molecule was generated. But the question is, now the second question is, how this gradient was created across the membrane? How the cell creates this gradient? I want to explain you. Cell performs specific mechanism. I am taking example of photophosphorylation. In this case, the inner membrane of thylakoid membrane. This is the inner thylakoid membrane, and we have a specific molecule, that is light harvesting complex associated with the reaction center. When the sunlight falls on it, photons falls on it, the electrons are excited. This electrons was trapped by another molecule, that is plastocyanin. Plastoquinone. That is plastoquinone. That is plastoquinone. When the electrons moved from this reaction center to plastoquinone, the protons are moved from matrix to inner thylakoid membrane. I'm showing you. This is the inner thylakoid membrane, and the protons are moving here when the electrons are transferred. So the high concentration of proton rises here. When this plastoquinone transfers electrons to another molecule, that is cytochrome, the electrons are again transferred and the protons are again being transferred. The cytochrome molecules transfer electrons to plastocyanin, and again the protons are being transferred. So the proton gradient was created again and again across the membrane. And what is going on? When this plastocyanin was transferred, electrons was transferred, the proton gradient concentration was rising here. And what we have? We have a specific molecule, that is ATPase, located here, and these protons, when comes back from high concentration to low, the ATP generation takes place. Because the electrons were generated with the help of photons, this mechanism of generation of formation of ATP was named as photophosphorylation. phosphorylation. So, because phosphorylation means addition of phosphate. So ADP plus phosphate means phosphorylation and formation of ATP. What is going on? In this way, in the same way, in case of mitochondria, what is going on? In case of mitochondria, the electrons was generated by the oxidation of glucose. These electrons were received by NADH, and ultimately this NADH transfers these electrons to the biomolecule, specific biomolecule I am showing here. Through a specific biomolecule, that is NADH ubiquinone oxidoreductase enzyme, and transfers electrons from NADH to ubiquinone. And when this happens, the proton gradient was created. Protons was transferred from matrix to inner membrane. Again, what we are observing in the same way as in case of chloroplast, the gradient was created across the membrane. This ubiquinone transfers electrons to the cytochrome, again present in the membrane. Same thing happens, the gradient was created. When this moves to the cytochrome C and then again to cytochrome oxidase, the proton gradient was created. Again, a specific molecule that is also named as F1F0 particle. It is an enzyme, ATPase, and this, when this H moves back through this ATPase enzyme, the ATP was generated. In this case, the electrons was obtained by the oxidation of glucose. So, this mechanism was named as oxidative phosphorylation. So, my dear students, here we observe that how the cell is generating ATP. But the question is again that ATP is generated. How the cell performs its activities by its own? How? So I'm explaining you with a simple example. Glucose. We know it is an energy molecule, but the energy cannot be created outside a cell. The energy can be created only inside a cell. How? Because we know that glucose cannot disintegrate by its own and we cannot generate energy. We have, because the first chemical reaction of this glucose catabolism, that is glucose plus phosphate to glucose 6-phosphate, is a non-spontaneous reaction because the delta G is plus 20 kilojoule per mole. This is a non-spontaneous reaction. This is not feasible. So that's why the glucose catabolism cannot take place by its own. So what do the cell do to catabolize glucose? First of the cell, to make it feasible, what the cell do? So I'm writing here the another reaction, that is ATP hydrolysis. This reaction is highly spontaneous at its delta G is minus 30 kilojoule per mole. What do the cell do? Cell just couple these two reactions. And the now reaction is written as and the delta G, by the simple law of Hess, you can calculate it is minus 10 kilojoule per mole. So this non-spontaneous reaction was made spontaneous in a living cell by just coupling it with a high energy phosphate hydrolysis reactions. So the cell perform its all the reactions, all anabolic, all the type of reactions which are not feasible outside a cell are made feasible by coupling it with a high energy phosphate hydrolysis. And the phosphate generation was take place by creation of proton gradient across the membrane. So this is the mechanism, this is the simple glimpse that how the cell generates energy and utilize it to make a non-spontaneous reaction spontaneous and creates energy for performing its all the biological activities for by its own. I am giving you a simple example of the bacterial movement, that how you can observe. This is the bacterial cell. This is the inner membrane, and when the proton gradient was created across the membrane, and this ATPase enzyme through enzymes move and it rotates clockwise. This ATPase enzyme was linked with a specific protein MOT and to a specific molecule, that is a flagellum. So we can just conceptualize it by a screwdriver. When we rotate this screw clockwise, what happens? It moves forward, and when this proton passes through this ATPase molecule, the ATPase molecules rotate and it was linked to a specific molecule, that is flagellum, which rotates like a screw. What will happen? The bacteria will move forward. The electron generates from where? When there is a glucose supply here, and how the bacteria moves, performs a chemotactic movement. It moves in the direction of the food and nutrition, because the proton gradient was created on that side where there is a food, and the bacteria moves towards the chemical molecules. So in this way, we can understand how a simple mechanism of creation of gradient across a membrane can help, is making a cell, a non-living cell, into a living form. So today we understand how the cell creates energy, generates energy, and utilizes it to perform its all the biological activities. We will meet again and extend our lecture about biomolecules and certain facts of bioenergetics, which will help us to better understand the phenomena of creation of energy and the generation and utilization of energy in the cell. Okay, thank you.